We're alive now. Good afternoon, students. It's about 4:25, Monday, May 4th, and some of the stores are open today. So I didn't go out. I didn't venture out, but I have been told they're open. And hopefully everybody is staying safe and taking all their precautions, please. Remember, Thursday is your final exam, 9:30 in the morning. So you'll turn on to Final Exam Susan Angel, and hopefully I'll get it posted at 9:30 and you can take that exam. I am still going over vocabulary words with you, so let's focus in on what is next. And I think we discussed the postcentral gyrus and its function, and now Broca's area. If you remember, Broca's area was part of the brain, a location in the left hemisphere of the brain, an area about the size of half a dollar or a silver dollar. Usually it's in the left side of the brain, the left hemisphere, lower back from the frontal lobe, maybe into the parietal lobe, but it's usually located right there on the left side. It sometimes in individuals can be found on the right side, but you don't have two Broca's area. You just have one, mostly the left, sometimes in the right. And the Broca's area was an area in the brain that a doctor by the name of Broca in England discovered. When someone has had a stroke or brain damage in that area, then it hinders speech. And someone who's had a stroke, they may be thinking correctly and trying to talk to you and communicate, but it comes out as gibberish. It's very frustrating to them because they know what they want to say. They just can't say it. And sometimes they'll try and write it out, or if they have a tablet, they might be able to print out what they want to communicate. But that's Broca's area, and it was named after Dr. Broca, who was the medical biologist that discovered the significance of that area. Broca's area. Now, the next was brain lobes, location and function. And if you can remember correctly, one of the first things we learned were these lobes. What is in pink here is the frontal lobe. It is separated by the purple parietal lobe, by the central sulcus. That's one of our brain landmarks. And if you remember correctly, the tissue right in front of the central sulcus, the precentral gyrus, that area was significant in gross and fine motor movements. Right behind the central sulcus, in the postcentral gyrus, is the area that's responsible for our awareness of tactile sensations. The frontal lobe is separated on both sides by the central sulcus, and it's separated from the lower temporal lobe by the lateral sulcus right here. That's the lateral sulcus. Now, in purple was the parietal lobe, frontal, parietal, and then right behind in the latter portion of the skull, illustrated in a darker blue, kind of a cobalt blue by the artist, that is the occipital lobe, which is responsible for sight perception. Remember, you see with your eyes, which bend the light to fall directly on the fovea centralis in the retina. The message is carried by those optic afferent sensory neurons through the optic disc into the optic nerve. It forms the chiasma and then notifies each side, each lobe of the occipital lobes, what you're seeing. So if you cross or if you look at something with your left eye and you can't see it, you've bandaged the right, then that chiasma sends those messages of sight to both sides of the brain, and the same with the opposite. So When you're looking at someone with one eye, you're actually seeing him or her with both sides of that occipital lobe. The temporal lobe, which is here in kind of a light baby blue, that's responsible for our sense of sound perception. And remember, all of these are sent to the right area of the brain by the thalamus, which is part of the diencephalon. Remember, it's the traffic cop that sends everything to the right portion of the brain, which is coming up from the brain stem or going down the brain stem or utilized within the brain itself. That thalamus does that. Remember, everything except olfaction, the sense of smell, which goes straight to the brain, deep within the brain. So those are the brain lobes. As far as the frontal lobe, I do want you to remember that analyzation and thinking and memory is done right in this area. You really, it's where you are you. It's your personality. You think with your whole brain because your association neurons link everything you see, hear, taste, touch, smell, and feel so that you can make sense of your environment. Each lobe has a distinct function, but you utilize the entire brain to think. Okay. The next was the basal nuclei. We have mentioned that, and I looked for the diagram of the basal nuclei, and I do have it. Here is, turn it sideways so you can see it. Here is the brain, and what we've done is a transverse section. We've cut off the top, kind of like a jewelry case. You just lift the lid, and you can see the white matter in the center of the brain, which illustrates neurons that are heavily myelinated. And in some areas, in these 4 distinct areas, you see these areas are referred to as the basal nuclei. These clumps of cell bodies are responsible for receiving the impulses from the precentral gyrus, which controls gross and fine motor movements. The impulses go to the basal nuclei, and the basal nuclei then send acetylcholine, use acetylcholine as the transmitter to the thalamus and down the brainstem so that it controls your gross and fine motor movements. They do go through the basal nuclei that regulate them. And you can see, I like this one the best. That's my favorite picture of it because I think it's so easy to distinguish. But if you look in your notes, and I wish they had used this picture, but if you look in your notes on page 405, figure 1110, you'll see an illustration of that basal nuclei, and they show a real brain in cross-section in which you can see the darkened areas. And it's labeled there, but that is my favorite because I think it's so very definitive. Basal nuclei. Remember, when you have a clump of cell bodies in the central nervous system, it is referred to as a nuclei. Now, we use that nuclei a lot in science because we talk about the nucleus of an atom in which the protons and the neutrons clump together. We talk about a nuclei in a cell, which contains the DNA and the RNA, or the directions for that cell. And again, we talk about in neurology a nuclei, which is just nothing more than a clump of cell bodies. If you take the same type or clump and put it in the peripheral nervous system, then you have a ganglia. And we talked about the dorsal root ganglia. We talked about the sympathetic chain or the lateral ganglia or the paravertebral ganglia, and the collateral ganglia and the terminal ganglia. So any clump of cell bodies outside the brain and cord is a ganglia. Inside, it is a nuclei. Ependymal cells, I mentioned these. These are very special neuroglia cells located in the central nervous system. These little cells have cilia, and they're significant because they exist in the choroid plexus of the first and second and third ventricle and the fourth ventricle. And you also find these ependymal cells, these neuroglia, are also found in the central cord, the central canal of the spinal cord. And again, what they do is they're significant in extracting from the plasma that nutritive cerebral spinal fluid, which is only about 6 to 8 ounces and flows around the brain through the ventricles and down through the central canal of the spinal cord, as well as around the entire cord. So the ependymal cells are significant in forming that. The astrocytes, we mentioned it yesterday, and I did point out a picture in your textbook. They're a form of neuroglia that exist in the brain, and they wrap around capillaries, and they form part of the blood-brain barrier, which develops at the end of the first year of life. And it inhibits some amino acids and other toxins from leaving the capillary by that hydrostatic pressure and entering or bathing the cells of the brain. There are some substances in the capillaries, in your blood, they're fine in your body tissues, but they're not to be exposed to brain cells. And so it's the astrocytes that wrap around those capillaries and help provide that blood-brain barrier. And the blood-brain barrier is also worked because the capillary membrane has very tight junctions. Things do get through that need to feed the brain cells, but it also works to inhibit some of these toxins and some of the amino acids. So the tight-fitting cell membrane of the brain capillaries work synergistically with the astrocytes in providing that blood-brain barrier. Remember, it does not exist until the end of the first year of life. So when mother is carrying the fetus or when the baby's in its first year of life, whatever she eats can go, or whatever she drinks, or if she takes medication, or God forbid, drugs, it can go directly into that baby's brain, causing disabilities and sometimes gross. Fetal alcohol syndrome is a good example of that. Mother's an alcoholic, drinks a great deal every day while she's gestating, and the alcohol is not inhibited. It goes into the baby's brain and can cause retardation and so lots of physical abnormalities. Okay, but I went off on that, but that's the ependymal cells and astrocytes. Cerebral spinal fluid, all that means is that 6 to 8 ounces of fluid that comes from your plasma and is taken into the ventricles and around the brain and around the cord and down the central canal and forms what we call the cerebral spinal fluid, which is extremely nutritive. It is filtered from the plasma by the ependymal cells. Okay. Now, the cerebral spinal fluid is what they may tap. They'll do a puncture and extract some of that fluid when they want to see if a patient is suffering from encephalitis or meningitis, viral or bacterial meningitis. They'll do a spinal puncture and extract that. Now, one thing you did have to know is the passage of the cerebral spinal fluid through the brain, and that's beautifully illustrated in your text. So I am referring you to that. And I'm also going to go over this a little bit with this transparency. Now, you can't see this, but this is the inside of the thalamus. It represents the third ventricle. And in the third ventricle, you do see the artist has illustrated a little bit of the choroid plexus, which contains the ependymal cells. The fluid goes into the third ventricle. It drains from the third I'm sorry. I'm talking about the third ventricle. Let me back up and start up to the lateral ventricles. You have two lateral ventricles, one in each hemisphere. And in those lateral ventricles, you do have the choroid plexus, which picks up on the cerebral spinal fluid from the plasma. It flows from the first and second lateral ventricle to the third lateral ventricle by the interventricular foramen. Once it gets into the third ventricle, which is surrounded by the thalamus, it flows from the third ventricle to the fourth ventricle by the interventricular foramen, and then on down. Let me back that up. First and second to the third by the interventricular foramen, third to the fourth by the cerebral aqueduct. Thank you. And from here, it flows, as you can see, all the way around the brain, all around the cord, and also through the central canal. So let me reiterate, because I probably confused you. It forms first in the two lateral ventricles, one on each side in each hemisphere. The fluid flows from the first and second lateral ventricles into the third ventricle by the interventricular foramen, from the third to the fourth by the cerebral aqueduct, and then around the brain and around the cord and through the central canal. And I'm sorry I stumbled through that, because I'm glad I caught it, because I didn't want to give you any misinformation. Okay. The brain stem. Now, remember we talked about the brain stem yesterday or day before yesterday. The brain stem, some anatomists just simply consider the midbrain, pons, and the medulla oblongata the brain stem. And others say, no, we'll incorporate the diencephalon, which is the thalamus and the hypothalamus, the infundibulum, and the posterior pituitary. So I do mention to you that there's 2 points of view. You know what the thalamus does. It directs all information to the right portion of the brain. The midbrain, which in a mid-sagittal section kind of looks like a duck's head. And the midbrain is important because it relays impulses from the upper brain and the thalamus down through to the pons, medulla oblongata. But it also is responsible for eye and ear reflexes. If a loud noise happens in a room to your right, somebody drops a book, you turn to the right. You don't think about it. That's an autonomic reflex. You turn to the right, you look to the right autonomically in the area of the sound. That's eye and ear reflexes. Below that, it kind of looks like a bubbling out of tissue in the brainstem right in front of the cerebellum. This is the pons. The pons connects the midbrain to the medulla oblongata and also acts to kind of coordinate both hemispheres of the cerebellum. Below the pons is the medulla oblongata. It's a very important area because there are nuclei in the medulla oblongata that take care of respirations and maintain respirations depending on the saturation levels in your blood of oxygen. And if your oxygen sats go down, then you increase respirations directed by the medulla to pick up on the respirations to increase and bring the oxygen levels back up. It's also significant in controlling the smooth muscles of your arteries, arterioles, and helping to maintain proper blood pressure, the constriction of those smooth muscles in the walls of those blood vessels to maintain hydrostatic pressure and blood pressure. And it's also involved with cardiac, which it regulates the number of strokes or contractions your heart gives in one movement or in one moment. That's the medulla oblongata. And if injury occurs to that medulla oblongata, for example, Christopher Reeves, when he took that spill off a horse, he injured. He was the first Superman. Well, not the one in the 50s, but the one in the 70s or 80s. Anyway, he injured the respiratory center in the medulla oblongata, and he had to be on a respirator. Okay, that is the brain stem. The limbic system is a system that we didn't know a whole lot about. I do have a little transparency here for you. The limbic system is a number of scattered neurons linked together that surround the diencephalon and the brain stem. The limbic system is a system that's extremely important in emotional development and emotional expression. You've heard of people that are terribly histrionic or very, very emotional. Then you can believe that the limbic system is extremely sensitive and that it's expressed that way because of the sensitivity of their limbics. There's not a whole lot known about it, but we do know that it's involved in emotional expression. Reticular formation. The reticular formation is a number of neurons that come up through the brain stem and surround the brain stem and the thalamus. And the reticular formation is extremely important in sifting what is significant information from insignificant. For example, if you're listening carefully to the video and you're trying to concentrate on these neuro vocabulary terms, then there's lots of other things going around in the environment. The air conditioner may be turning on and off. I'm looking into the camera, and behind me is a window, and the breeze is blowing through the leaves, and there's a lot of movements. But what the limbic system, what the reticular formation does, is it enables you to ignore surrounding stimuli and focus on what is significant. Okay, there are some brain injuries. They believe that autism may be one example or incidence of neural problems in which the patient cannot adequately sift information that's important from unimportant information. It all seems important to them, and they have what is known as a sensory overload. But that reticular formation, if you remember it, it sifts what is significant from what is insignificant for the individual and which is important. Okay, that's about 23 minutes. Tomorrow, I'll go over some more words. That's Tuesday. And if you have any questions, again, please call me. I want everybody ready to know these terms by Thursday morning. So please call me if you have any questions about any of these terms. Your textbook is a great source. So you can look up the terminology in your textbook, in your class notes again, and you can even Google terms. You can Google reticular formation or basal nuclei, and that is a source too. But any questions, please call me. I miss you all, and if you're starting to go out there because some of the stores are open, don't be lax. Take care. Wear your masks, and don't touch your hands to your face when you come home immediately. Scrub those hands, and try and disinfect or put your mask in ultraviolet light. Just 30 minutes of ultraviolet light is destroying the membrane around that COVID-19 virus. So stay healthy. I don't want any of you to be hurt from all this mess. And thank you. Appreciate it. Sure do miss you guys.